Hi Year 10s, welcome to week 3 of biology in Year 10. This week we're going to be looking at genetic modification. This is, like I said, our third part of the 10-week course. As per usual with our videos and our PowerPoints, this is the 3 criteria or the Australian curriculum standards that you need to be able to do at the end of this week. So at the end of this week you must know the history of DNA sequencing, you must know the applications of gene technology, and you must be able to talk about genetic testing used for embryo selection, carriers of genetic mutations, use of information by companies and medical authorities. Okay, so it's 3 nice easy points. But to start off, we need to talk about what is genetic modification. So genetic modification, this is the ways in which humans have basically learned to modify the DNA that's inside of cells to basically create characteristics that are of benefit to the human race. When those cells that have modified DNA go through cell division, every new cell that's created through that cell division has a copy of the genetic information, so they become cells themselves with modified DNA. So scientists have mostly modified plants to add desirable traits. So traits that we've added are things like resistance to insects, resistance to herbicides, so we can kill the weeds that are around those plants, and increased nutrient value. Genetic modification in itself, that's not exclusive to plants. We have done it to other animals. So in China, for example, they're using it to clone animals. There have been scientists that have made things like glow-in-the-dark fish. Okay, but the idea of genetic modification itself is highly controversial. Sometimes in science, so scientists agree that, okay, it's good that we have this ability, but there's a limit to what we should do. But then there's also the community debate of, should we be having genetic modification at all? So that's interesting. There's a lot of people that won't eat genetically modified food, for example. So in class, we'll watch this little video as well, which is what is genetic modification. I won't play this on here because I don't want to play someone else's YouTube video inside a YouTube video. I might put the link down below in the comments if you do want to watch that video, but we will show it in class as well. But let's talk about some examples of genetic modification. So we're going to look at 2 examples of how humans have genetically modified plants. So the first one's going to be canola plants. And canola plants, basically canola is grown to be used for oil and food preparation. So canola oil, you might have used it at home. And generally this is grown in Australia quite a bit, and farmers will spray herbicides throughout the crops to kill the weeds that grow around the canola crops. So a lot of weeds grow around canola crops. The problem is that herbicides will also kill and harm the canola, which means that farmers that want to get rid of the weeds, because the weeds are stealing the nutrients from the soil and stealing the resources that the canola needs to grow, to kill the weeds, they risk losing some of their stock as well, what they're going to sell. So in New South Wales, Western Australia and Victoria, the government passed some rules that farmers are actually allowed to grow a genetically modified version of the canola plants. And that genetic modification makes them resistant to herbicides. So you can grow canola, and you can spray the weeds with herbicides, but the canola won't die because it has a gene that makes it resistant to herbicides. There's lots of concerns across the community that these could lead to mass problems. So one of those problems that people are considering is that plants breed by cross-pollination, and the public is concerned that the weeds, the canola plant that has this herbicide resistance will breed with the weeds and will have a super breed of basically weeds that can't be eradicated with herbicides. But that's really unlikely to happen, generally because they are such different species that any breeding is very unlikely to result in offspring. So it's highly unlikely that the weeds will actually become genetically modified to be resistant to herbicides. What's a bigger of concern, and it is actually true, is, like I said before, there's a lot of concern people don't want to eat genetically modified foods. So there are this group of the community that don't want to buy genetically modified foods. So any canola oils that's made from genetically modified canola, they won't purchase. So you have these farmers that specifically, they carry something called the non-genetically modified food certification. So they sell food that's not genetically modified. Now, canola plants and farms are very close to each other. Wind can pick up the pollen and blow it across the farms, and a genetically modified pollen, or pollen from a genetically modified canola, could fertilize a non-genetically modified canola plant, and then all of a sudden the genetically modified traits are going to be in that offspring. And those farmers then end up with genetically modified crops even though they don't have it. This is a really big problem because most of the genetic modifications that happen to plants, they're actually copyright. So someone makes this new strain of a plant, and then they put some copyright onto it, or they basically license it to themselves. And then if another farmer grows it without paying them some fees, then they sue them. So there was actually a movie, I think last year, in 2019, that basically there was a farmer that was collecting his seeds every year to grow them. He was not using genetically modified plants. His plants had been pollinated through the wind by genetically modified plants, and then he was sued for using their seeds, even though he'd just collected it himself and it was just blown over by the wind and he didn't even know. So there's some problems definitely with this, but generally, in terms of consumption, it's safe. In terms of will it going to cross into other weeds and make herbicide-resistant weeds? Not very likely at all. So genetically modified canola is one that's quite safe. Another one that's a little bit of more use to the world currently, or could be of more use to the world currently, is golden rice. So rice is the main food source for around half the population of the world, probably slightly more than half the population of the world, because it's so easy to grow, it doesn't take up much space. So when we're looking at developing countries, rice is a really great food to grow and to eat because it doesn't take many resources. You can put lots of it in a small amount of space and feed a lot of people. But the problem is that white rice doesn't contain essential vitamins and the minerals that help sustain humans. So even though you're not going hungry, you're not getting the nutrients and vitamins that you require. So for example, one of those is vitamin A. So in developing countries, vitamin A can lead to a lack of vitamin A can lead to childhood blindness. So in developing countries where they're not actually having access, they're just eating white rice and they're not having access to vitamin A, there's a large population of children going blind. Now, to combat that, because we still want to use rice because it's so beneficial to use rice and it can help feed a large amount of people, scientists have been working on genetically modified rice. So they've taken genes from daffodils, corn, and bacteria, and they've added it to the rice. Now that genes that they've added help the rice contain something called the beta-carotene gene. Now you might have heard that because beta-carotene is basically what's in carrots and makes the carrot goes orange, hence why we're calling this golden rice. When we eat beta-carotene in our body converts beta-carotene into vitamin A. So it helps reduce that blindness because you're getting vitamin A in your body. Very important for developing countries. All of a sudden, we're getting vitamin A, we're reducing childhood blindness, which is going to really help the prospects of those children and eventually maybe help the prospects of the family. The problem is, unfortunately, yet it's not really been approved for human consumption. It's only allowed basically to be used by scientists to research, but they're pushing to get this out to the public, and hopefully this will help developing countries quite a bit. Now, again, another video that we were watching in class is about GMOs, and like I said, there's a lot of backlash in the community. Are GMOs bad? And the short answer, no, they're not. GMOs are really safe, and they're, in some senses, really useful for the environment. But this video will address, are GMOs bad? And we'll watch that in the classroom. So, gene splicing. Gene splicing is a way of genetic modification. Again, this is a really important one for humans because this is how we make insulin for diabetics. Gene splicing is when we take a gene from one organism and we place it into another organism, generally a bacterial cell. Generally bacteria is because they have a special section of DNA called a plasmid, DNA plasmids. We drew that when we did prokaryotic cells. Those plasmids are generally used for sharing genetic information between bacteria. They only replicate through binary fission. So every time they basically reproduce, you're getting a genetically identical copy of themselves. So there's no genetic variation. So they have this section of information of DNA that they can exchange with other bacteria to help increase that genetic variation and to combat viruses so they don't die out from viruses. We've then learned that we can remove sections of that plasmid and we can put our own genes in there and the bacteria will take the plasmid back up and then they'll be able to multiply and create that gene, or the products of that gene, the protein of that gene, in mass scale. So what we've done for bacteria with insulin, we've taken the human gene for insulin, we've spliced that into the DNA plasmid, we've let the bacteria take up the plasmid, and then we've let the bacteria replicate, and all of a sudden we have hundreds upon thousands upon millions of bacteria that can produce insulin for us, and then we can harvest that insulin, give it to diabetics. Much better than the way we were using it before, which was we were basically taking insulin from pigs. The problem were there was people who were allergic to it. We couldn't produce enough insulin to cover the amount of people that were diabetics, and there was this shortage, or lots of people that weren't getting treated for diabetes. Now, because we can create it on a mass scale, it's human DNA, there's a lot less complications, and a larger population that suffer from diabetes are being able to get treatment. Moving on, talking about the Human Genome Project. So this is a little bit of the history about basically when the human race decided we wanted to sequence every gene in the human body. So this was a collaboration across many countries, where basically all countries came together trying to identify the genes in the human body and determine the base sequence, so the order of the bases in the human DNA. Basically, this process lasted 13 years. It started in 1990, finished in 2003, when a rough draft was published. Now, while that was published in 2003, obviously you can imagine there's a lot of information. There's 46 chromosomes in the human body. Each one contains hundreds to thousands to millions of genes. So we're looking at millions to billions of genes here that we have to identify. Scientists are still going through and trying to figure out exactly what each gene does. We know the majority of them, but we're still trying to figure out what every gene does, so we can understand how to fight diseases, genetic diseases, and understand characteristics, etc. This process has actually been sped up a lot now. We can do this, we've been doing for other animals, and that process can essentially take days now instead of years. You can do it on a personal scale. You've got companies like 23andMe, I think there's one called DNA&Me, I'm not sure of that one. But basically you can give them a section of your DNA generated through saliva, and within a week or two they'll send you, here's your genetic information, here's the genetic disease you may suffer from based on the genes you have, etc., etc. And then there's a lot of other problems with that, which we'll discuss in class, such as who controls that information. It's your DNA, but that company now has a copy of your DNA stored somewhere. What happens if they get bought out by an insurance company? The insurance company sees that you have a chance of suffering from this genetic disease. Consequently, at that point, they're going to give you increased insurance costs. Might be employment opportunities that are gone because the information is available that you may suffer from this genetic disease later in life. Lots of complications with that, and there's no concrete laws really in place in Australia yet for that, so there's a lot of problem with those. But the fact is the technology is here that we can do this, and we're going through and we're making the genomes for many animals now to help understand how science and nature works in general. Leading on to that, because we now know where certain genes are in the body, we can do something called gene testing. Which is basically we do a test to see whether a person contains a specific gene. In Australia alone, there's more than 400 different genetic tests available to see if people have certain genes in their bodies. So for example, you can see if you're a high chance of heart disease or type 2 diabetes based on your genetics. That's really useful because if you find out there's a high chance you're going to suffer from heart disease or type 2 diabetes, then you can alter your lifestyle to reduce those risks so that hopefully you don't encounter them in your life. There's also other tests for more serious genetic diseases like cystic fibrosis and Huntington's disease. While some people might see that that information is dangerous to know, it is useful, for example, if you want to plan a family. So if you want to go ahead and plan a family, you want to see if you've known someone in your family has one of these genetic diseases, you want to see if you're a carrier and it might be given to your children, or to see if you're going to suffer from the effects of that disease early on in your life, and hence may not be able to care for your children effectively. It's important for those types of things, but then there's also people that if they find out they're going to have something like Huntington's disease, it kind of puts a halt on their life and they think, well, what's the point? So in some senses, they are negatives to knowing this information. So it's a really serious consideration of, do I take this test to figure out if I have it? How is it going to affect my life in the long run? The important thing with those tests as well is one, there can be false positives, so you can get the information back that says maybe you're going to have cystic fibrosis, or there's a chance you'll have cystic fibrosis or Huntington's disease, but it could be a false positive and you don't actually have that gene. There is always the chance of that happening. There's also the opposite. You could get the false negative telling you that you're not going to have those genes, even though you do. And the other big flaw with them is they'll only predict the chance of ever encountering that disease. They're never going to predict how severe your symptoms are going to be or what age you're going to see it at, just that you may or may not get it and what chances there is. So while these tests are available, there is still massive flaws with them. So a little bit, it kind of just talked about this slide in that last one. So genetic testing can negatively affect the well-being of an individual if they know they're going to suffer from a specific disease. Knowledge of a potential specific disease could increase insurance costs because they know there's a chance you're going to suffer from this later on, and consequently, they know they're going to be forking out a lot of money to help you go through the symptoms. Knowing a family member has that gene could cause concern to family members who are worried about that person. Could also cause concern to family members who may carry that disease, not knowing, and they don't want to be tested, but knowing someone else in their family has it knows they have an increased chance. So the well-being of individuals is really important when you're thinking about taking this genetic testing. Okay. Lastly, one that we're going to focus on a lot next week is that now that we have the ability to use genetic modification, we can actually use DNA to develop vaccines. So how vaccines work, they expose your body to a weakened or dead pathogen, cause your immune system to think it's infected even though it's not, and initiates a response by making antibodies, and your body will store some of those antibodies so that if you ever do encounter that virus, your body can produce those antibodies fast and fight off the infection before you ever see symptoms. That normally is a long process, but there are new types of vaccines that are being produced where we can take the DNA of the virus, recreate that DNA to make a protein, or to get your body to make a protein that recognizes that protein as a virus, and hence will fight off any infections because your body's seen the protein that's on the virus without being exposed to the virus. This specifically is how Pfizer vaccine was made for COVID, and we're going to look at that really a lot more in context and a lot more in depth in our Sciences to Human Endeavour task, which we're going to start once we finish these notes and in next week. Thanks for watching. See you in class.